Alright, in this video, we're going to look at osmosis and diffusion, and these are both passive processes by which substances can enter and leave the cell, so they require no energy. And I'll start with diffusion. So diffusion is the random movement of molecules from a region of their high concentration to a region of their low concentration down a diffusion gradient. Now, a diffusion gradient can be described as a difference in concentration between 2 points. For example, if it's perfume, one example of diffusion is if a perfume is sprayed on one side of the room, you realize that after a certain time, even the person on the opposite side of the room will be able to smell it. This is because the perfume molecules spread evenly throughout the room, and this occurs by random motion of molecules due to kinetic energy down a diffusion gradient. Now, diffusion in the body occurs across the cell membrane, and it's essential for many cell functions. Now, the factors that affect the rate of diffusion across the cell membrane are summarized by what is known as the Fick's law. And the first law states that the rate of diffusion is directly proportional to the surface area of the membrane. So the greater the surface area of the membrane through diffusion takes place, the greater the rate of diffusion. The second one states that the rate of diffusion is directly proportional to the difference in concentration or the concentration gradient across the membrane. And that basically means that the steeper the concentration gradient, the faster the rate of diffusion. Okay. Now, the third states that the rate of diffusion is inversely proportional to the distance over which diffusion takes place or the thickness of the membrane. So that means that the thinner the membrane, the greater the rate of diffusion, and vice versa. Now, other factors include temperature, size of diffusion molecules, and concentration. Now, diffusion is divided into two: simple diffusion and facilitated diffusion. And I'll start with simple diffusion. Now, this is the type of diffusion where small nonpolar molecules cross directly through the phospholipid bilayer of the cell membrane. Now, these small nonpolar molecules include respiratory gases such as oxygen and carbon dioxide. Now, some ions also undergo simple diffusion, only that they diffuse very slowly through the phospholipid bilayer. Now, water is polar, but it's small enough to cross the phospholipid bilayer, especially the hydrophobic region, without interference. And I hope you guys have got that one about simple diffusion. And then we come to facilitated diffusion. This is the type of diffusion where ions and polar molecules cross the phospholipid bilayer through special transport proteins. These are carrier proteins and channel proteins. And these facilitate the diffusion of some ions and polar molecules such as sodium ions, chloride ions, potassium ions, glucose, and amino acids. Now, that's why it's called facilitated diffusion. Now, there's a difference between channel proteins and carrier proteins. And channel proteins have a fixed shape and form water-filled channels across the cell membrane, and these allow passage of water-soluble ions through them. Now, these channels are selective, each opening only in the presence of a specific ion, so they exert some control over which substances can enter the cell. Then carrier proteins, these are selective and undergo a rapid change in shape when a particular molecule specific to that protein presents itself. The molecule binds to the protein at a specific binding site and causes it to change shape in such a way that the molecule is released either inside or outside the cell depending on its concentration gradient. So that is all on diffusion. Now, if I've missed anything, you guys are very free to do your research so you can get more details on diffusion. Now, we now turn our attention to osmosis. Now, this is just like diffusion, but it involves solvent molecules like water. Now, osmosis, actually, before I give the definition, there are terms I need you guys to come to relate with because they'll come in handy as we go on. And first is water potential. Now, this is the tendency of water to move from one area to another. And one thing you should know is that water moves from a region of high water potential to a region of low water potential. That will help you a lot. Pure water has the highest water potential of 0, so it's usually negative. And as you add a solid, maybe salt, to pure water, its water potential becomes more negative, so it reduces, and that is called solid potential or osmotic potential. So solid potential is therefore the measure in change of water potential due to the presence of solid molecules, and solid potential is also always negative. So osmosis is the movement of water molecules from a region of high water potential to a region of low water potential. Note that the more negative the water potential, the lower the water potential, and the less negative the water potential, the higher the water potential. If we have our three cells with water potentials at negative 300 kilopascals, negative 500 kilopascals, and negative 800 kilopascals, the direction of water here will depend on their respective water potentials until we move from a cell of high water potential to a cell of low water potential, as you can see. So from here you can notice that water moves down a water potential gradient. This an animal cells, and for this one I'll use a red blood cell. Now imagine a red blood cell is put in each of these solutions: a hypertonic solution, this is a concentrated solution, a hypertonic solution or a dilute solution, and an isotonic solution. Now, if a red blood cell is put in a hypertonic solution, the red blood cell will lose water to the hypertonic solution and it will shrink or crenate. Now, this is because the red blood cell has a high water potential compared to a hypertonic solution. If put in an isotonic solution, the red blood cell will not lose any water, so there will be no change because they both have the same water potentials. And if it's put in a hypotonic solution, the red blood cell here will gain water from the solution, and since its cell membrane cannot stretch to a very great extent, the cell will therefore burst, releasing its content, and this process is called lysis. Now, this happens because hypotonic solution has a high water potential compared to our red blood cell. So that's what you need to know on osmosis and animal cells. When it comes to osmosis and plant cells, the term protoplast is used a lot, and the protoplast is basically the living parts of the cell excluding the cell wall. So these are surrounded by the cell wall. Now, when a plant cell is put in a hypotonic solution, these have a higher water potential compared to the cell. Plant cell therefore takes in water by osmosis. Now, unlike animal cells, the plant cell does not burst due to its rigid cell wall. So the protoplast, remember, its volume increases until it starts to exert a pressure on the cell wall, and the cell wall responds by exerting an equal and opposite pressure on the protoplast, and this is where the term pressure potential comes in. Pressure potential is the backward pressure the cell wall exerts on the protoplast to oppose the continuous osmotic intake of water. At this point, the cell is said to be turgid. When a plant cell is put in an isotonic solution, I talked about this, there's no change because they both have the same water potential, so there's no net movement of water. Now, when a plant cell is put in a hypertonic solution or a concentrated solution, these have a lower water potential compared to the cell, so the cell loses water to the solution, and this causes the protoplast to shrink and it starts to pull away from the cell wall, and this is called plasmolysis. So the cell here is plasmolysed. And with that said, you can now turn our attention to this graph. This graph shows the relationship between water potential, pressure potential, and solid potential in a plant cell. Now, at full plasmolysis, as you can see, water potential is low or more negative due to the high amounts of solids in the plant cell. Now, the higher the amount of solids, the more negative or the lower the solid potential. As solid concentration decreases, the solid potential becomes less negative or it increases, and the water potential due to the low amounts of solids also becomes less negative and increases up to full Tagger. Now, at full Tagger, the water potential is 0, and that represents pure water. I'm not telling you guys to copy exactly what I've written here because this is just my summary of the graph so that you guys can get the point around this whole graph. Now, the pressure potential starts from 0 and increases to more positive values. This pressure potential can also be called Tagger pressure, and it starts from 0 because as water enters the cell, it causes the protoplast to exert a pressure on the cell wall, and the cell wall also creates an equal and opposite pressure against the protoplast, and that is where the Tagger pressure comes in, or our pressure potential. So as the water potential increases due to more water entering the cell, the pressure potential also increases to oppose the continuous intake of water. So at full Tagger, water potential is 0 and the solid potential is equal to pressure potential. This is everything you need to know on osmosis and diffusion. Now, understanding osmosis is very essential because it lays the foundation for you to understand so many biology concepts. So please like the video, subscribe to my channel, and hit that bell icon to get notifications every time I release new content.